I am Vaibhav from Vaibhav E-Learning Academy. Today we are going to start a new topic in biology, that is biomolecules. This is our part number 1. In today's video, we will be learning about introduction to what is biomolecules, what is the analysis of a chemical compound, analysis of inorganic compounds, primary and secondary metabolites. This will be our components in the first video. So let us start with the topic of biomolecules. Now, if you look at the term biomolecules, what does this term define? So when we do a breakdown of the word biomolecules, we see bio means biological and molecules are the components. So from starting we all know that from a cell we get tissue, group of tissues, from organ we get organ system, then we get an organism. Now in this chapter of biomolecules we will be learning what forms a cell. What forms a cell? These biomolecules form the cell. Not only biomolecules plus some inorganic substances form this cell. These things form cell, which forms tissue, organ system to the organism. This was the basic introduction to what is biomolecules. Now, what is the definition or what are biomolecules? The carbon containing molecules are known as biomolecules. The carbon containing molecules which are present in a biological system, for example the earth crust and the human body, so these are known as biomolecules. The carbon containing molecules are carbon containing components. Now after looking at a cell, how will you identify what are the things which are present inside a cell? To identify what are the things which are present inside a cell, we have to do an analysis. How will you do that analysis? There are some steps of doing an analysis. So let us look at these steps one by one. So now we are going to analyze what is present in a living component. Analysis of chemical compounds. So for this we have to perform some steps. What are the steps? So let us talk about theoretical things, what actually we need to do. For this we have to take a piece of a living cell. Suppose a leaf cell, suppose a plant cell, suppose a vegetable cell, or a piece of liver cells. We have to take that liver cell, we have to put it in mortar and pestle. What is mortar and pestle? This system is known as a mortar and a pestle, in which we have a small box like thing over here and with a stick on the top of which, by which we used to grind it. So this is known as mortar and pestle. So what we have to do is we have to take a liver cell or we have to take a vegetable cell. Some cells we have to put it inside the mortar and pestle. Then we need to add trichloroacetic acid. Trichloroacetic acid, which is Cl3CCOOH. This is a compound which we have to add in the mortar and pestle. Then we have to grind it. After grinding it, we have to make a slurry. Slurry in the sense that some sliming component. So we take a piece of cell, we add it in mortar and pestle, then we take trichloroacetic acid, we add it inside the pestle and then we grind it. After grinding, we obtain a slurry. That slurry, we take it and we keep it on a paper, a cotton paper or a cheesecloth. After putting it on cheesecloth, we have to press it or we have to squeeze it, then we will get two components. After doing that analysis, we get two components. We'll be looking into the components, but first let us write down the steps what we have performed. So the first one is take a living cell. This cell can be a vegetable cell or a liver cell. Then put it in mortar and pestle, and then add trichloroacetic acid. After adding this, we have to grind it. After grinding it, we have to filter through cheesecloth. What is a cheesecloth? A cheesecloth is a cloth which has some small pores in it, very minute pores, through which some soluble substance can come down and insoluble substance will remain at the top. After filtering through cheesecloth, we will get two components. Now what are the two components? One component is known as filtrate and the other component is known as retentate. So after the filtration, we get two parts. One part is known as filtrate and one part is known as retentate. Now what is filtrate and what is retentate? Filtrate is the thing which comes down after you squeeze it through the paper, or else after you squeeze it through the cheesecloth, the liquid which comes down or the substance which comes down is known as filtrate. This filtrate is also known as acid soluble pool, or these are known as biomicromolecules. What are biomicromolecules? These biomicromolecules are also known as biomolecules. So this biomicromolecule, also known as biomolecules, also known as acid soluble pool. What is it? Why is it soluble? If it is soluble, because it is soluble, it is coming down in a mixture. It is coming in filtrate, and the opposite will be acid insoluble pool. Insoluble pool. What is pool? Pool means the mixture, the acid soluble mixture and acid insoluble mixture. This retentate is the thing which remains in the cheesecloth, and filtrate is the thing which comes out of the cheesecloth. This acid insoluble pool is also known as biomacromolecules. So here we have two components. One is biomicromolecules, which are known as biomolecules, and then we have biomacromolecules. Now this is a thing which comes out of cheesecloth, and here it is remains in cheesecloth. Now the examples of biomacromolecules. To understand this, we have to learn about some substances. For example, this is known as a carbohydrate, and over here we are talking about a polysaccharide. What is a polysaccharide? In our nutrients video, we have learned that a compound, over here carbohydrate, a polysaccharide is made up of very small monomers. This carbohydrate is made up of glucose plus glucose plus glucose. So this is a sugar, carbohydrate, this is a sugar, polysaccharide sugar, and these are the small components. So here we have one glucose, another glucose, so there are n number of glucose which combine to form one sugar. So over here, this total carbohydrate is a retentate because it is big, and glucose, that is simple sugar, will come under filtrate. So here example is simple sugar, and over here we will have polysaccharides. Okay, this was about simple sugar. Now similarly, let us talk about another compound, that is protein. Proteins are made up of very small amino acids. So we know that there are 20 types of proteinaceous amino acids. So these amino acids will combine in some different proportions to form a protein. Over here, protein is a large molecule, so protein will come over here. And over here, amino acids are the small monomers which will make the protein. So over here you will get amino acids. After protein, let us suppose nucleic acids. What are nucleic acids? The components which form the DNA and the RNA. So nucleic acid, for example, this is a big component. This big component will be made up of many small components, which is monomers, which are called nucleotides. What is nucleotide? Nucleotides are the monomers which form the nucleic acid. So over here, nucleic acid is the large part which will come over here. And over here we will have small parts, that is nucleotide. So this was the basic difference between filtrate and retentate. But over here there is one component missing, fats. Now this is a very exceptional case. So the exceptional case of lipids, this will be discussed in the later part of this video. Now one more topic about filtrate is that they are small in size. That is, they are less than 800 daltons. Dalton is a unit for measurement of the size of these biomolecules, and these are large in size. They are greater than 10,000 daltons, or we can say between 1,000 to 10,000 or greater than 10,000 daltons, which are known as macromolecules. So less than 800 are filtrate and more than 10,000 are retentate. This was the basic difference between acid and retentate. Let us revise. So what we did was, we took a piece of a cell, living cell, either vegetable cell, plant cell, liver cell or any other cells. We took that, we kept it in a mortar and pestle, we added trichloroacetic acid and then we grinded it. We will obtain a slurry. That slurry, we pass it through a cheesecloth. After passing through cheesecloth, you will get two components. One is known as filtrate, which comes down, and one is known as retentate, which remains in the cheesecloth. Filtrate is also known as acid soluble pool or biomicromolecules. This is also known as biomolecules. Then we have retentate. Retentate are acid soluble pool or biomacromolecules. Now these will remain in cheesecloth and this will come out of the cheesecloth. Examples are simple sugar, amino acids, nucleotides. And then we have polysaccharides, proteins and nucleic acids in retentate. This filtrate will have small in size. They are small in size, of course, because that is the reason why they are able to come out of the cheesecloth. And then we have the retentate, which is large in size, which is more than 10,000 daltons. They will remain in the cheesecloth and they are known as retentates. Now let us discuss about the strange case of lipids. So now we have discussed about the differences between a retentate and the differences between retentate and filtrate. Now in filtrate we have discussed about nucleotides, amino acids, simple sugar, glucose. In retentate we have discussed about polysaccharides, that is complex sugar, proteins and nucleic acids. Here we did not talk about lipids. Lipids are the fat. Why are we not talking about fat? So there is an exception in the fat. First of all, fats, they come under biomicromolecules, but not strictly. Suppose they ask a question that lipids are what, and in the option if they give both biomicromolecules and then biomicromolecules, we will preferably go with biomicromolecules. Why? Because they have, their size is less than 800 daltons. Their size is less than 800 daltons, then they are supposed to be biomicromolecules, but they come under acid insoluble pool. So what do you mean by this? So when we took that slurry and then when we filtered the slurry through the cheesecloth, we found that even though the size of lipid is less than 800 daltons, they are still remaining under acid insoluble pool. That is, they do not get filtered. They do not get filtered. So why do they do not get filtered? It is because they come under acid insoluble pool. But it does not mean that they have a small size. Even though their size is less than 800 Dalton, they come under acid insoluble pool. So that is why this lipid comes in a strange case. Exceptionally, we take it under biomicromolecules, but they are not under biomicromolecules or under biomacromolecules. This was about strange case of lipid. Now moving on to the next video, next part of this video, which is how will you identify the inorganic substances. So inorganic substances test. Now the test which we did right over here, this is known as the test for finding out the biomolecules. That is the test for finding out carbon containing molecules. But when you look about the cells, which I had written about here, cells make tissue. But here over cell, I have written here that biomolecules plus inorganic substances, Bm plus Is makes the cell. So for finding biomicromolecules, we did that test, that slurry thing. But for finding inorganic substance test, we need to do one analysis which is known as ash analysis. What is this ash analysis? Ash, as the term suggests, ash means after burning something. Analysis means we are learning about the substance which are present inside the ash. So for this, what we have to do? Now for this thing, again we have to take a piece of a living cell. Suppose plant cell, suppose vegetable cell or a liver cell. This time we do not have to grind it. This time what we have to do is we have to burn it. So first we take that cell and we heat it a little bit, burning it. So what happens is all the water contents, see actually what is a cell? As we said, cell is made up of biomolecules plus inorganic substances. Of course there is some water also, right? So first of all what we do is we take that cell, we burn the cell. Not burn, we actually heat the cell. So because of that what happens is water gets eliminated through evaporation. Now the remaining part is biomolecule plus inorganic substance. So after getting, see, before the cell was heated, before the cell was burned, that is known as wet weight. After we burn it, after the water gets escaped, that weight is known as dry weight. The dry weight does not contain water. Dry weight contains only biomolecules plus inorganic substances. Now we burn the cell. We burn it, like we put it on fire. Because of that what happens is biomolecules get erased. How? See, biomolecules are the compounds which contain carbon. When these carbon containing molecules or substances, when you burn them, they will burn and they will form carbon dioxide plus oxygen gas. So the biomolecules will get dissociated into the atmosphere. The remaining will be inorganic substances. What is the remaining? When you burn something, ash gets remained. So that ash will contain only inorganic substances. Analyzing these inorganic substances will give us the components which are present inside the cell. So let us write down what are the steps in this ash analysis. In ash analysis, we again take a living cell. This living cell, if we weight it, this is known as wet weight. What we do is we dry it or heat it. So after drying it or heating it, water is removed by evaporation. This weight is known as dry weight. This is known as dry weight. After this what we do is we burn it. Then ash is remained. So when we burn it, what happens is biomolecules, which is carbon containing, gets removed and then ash is formed. Study of this ash tells us about the inorganic substances which are present inside the cell. So this was the inorganic substance test, which is known as ASH analysis. We take a living cell, we weight it, that is known as wet weight. After drying it, the water gets removed. After burning it, the biomolecules get removed. When water is removed, it is called dry weight. When biomolecules are removed, it is called known as ash. And then when we analyze that ash, we find some inorganic substances. What are inorganic substances? Suppose sulfur, and then we have potassium, calcium. The substances which do not contain carbon in them are known as inorganic substances. So this was about inorganic substances test. Now we will be doing a weird comparison between the molecules or the components which are present inside the Earth's crust and which are present in human body with a table. So let us see. Before moving on to the comparison between Earth's crust and human body, we need to understand first what is present inside the human body. So now we did the first test, which is known as that slurry test. So in that, what we did was we took that slurry, we kept it in a cheesecloth, we filtered it, and we got retentate and filtrate. Now after looking at that, we will find what is the percentage of the components present inside the cell. So here we have component, and here we have weight percentage. First one, we have water. Water is 70%. We all know that 70% of our body is composed of water, but not only body, each and every cell of the body has 70% of water present in it. After water, we have proteins, which is 10 to 15%. Now, why did protein come? It is because we know that in the cell video we have learned about chromatin and chromosomes. So chromatin, that is chromosome. Chromosome is made up of chromatins. Chromatins are made up of small DNAs, and the DNA has small components which are known as genes. And every component or every substance which gives you about genetic information, that is made up of protein. So that is why protein is 10 to 15%. Even the most of the components of the cell are made up of protein. Ribosomes produce protein inside a cell. RER also produce, rough endoplasmic reticulum produce proteins. Even mitochondria and chloroplast in cells produce protein. So proteins are also present in good quantity inside the cell. After proteins, we have carbohydrates. Carbohydrates are present 7 to 10%. Carbohydrates are present 7 to 10% in our body. Sorry, carbohydrates are less in amount. Carbohydrates are only 3%. Why are carbohydrates only 3%? We will be discussing about comparison between the Earth's crust and human body about the some elements. So the first element which we have over here is hydrogen. So hydrogen is 0.14% in the Earth's crust, and it is 0.5 in human body. Carbon is 0.03, that is very little in Earth crust, but it is 18.5% in the human body. Then we have oxygen. Oxygen is 46.6% in Earth's crust and 65% in the human body. Nitrogen is very little in Earth's crust, but is 3.3% in human body. Sulfur is 0.3% in Earth's crust and 0.03 in Earth's crust and 0.3 in the human body. Sodium is 2.8% in Earth's crust and 0.2% in the human body. Calcium is 3.6% in the Earth's crust and 0.1% in the human body. Magnesium is 2.1% in the Earth's crust. Silicon is 27.7% in the Earth's crust, and silicon is negligible in the human body. And here magnesium is 0.1% in the human body. So this is the percentage weight present in the Earth's crust and human body, and these are the elements which are present inside the human body. So now we see that silicon, magnesium, calcium, sodium, sulfur, nitrogen, methyl sulfur, these all are the inorganic compounds, and hydrogen, carbon, oxygen, nitrogen, these are the organic compounds. So organic in the sense, organic means carbon containing, but these are not present inside the human body, not hydrogen, carbon, oxygen, nitrogen. C, H, O and N. So these are, C, H, O, N. These are the substances which are present in good amount in a human body, that is maximum amount in human bodies. So now, and that was organic, not inorganic. So now from this table, what did we find? We found out that all the elements which are present in the earth's crust are present in the human body. From hydrogen to silicon, the things which are present in the earth's crust, these things are present in the human body. Silicon is actually negligible in human body because silicon is not required in the body at all. Now, if we look at hydrogen, hydrogen is present in earth's crust very little amount, but human body in a good amount. Carbon is very little in earth's crust, but 18.5% in human body. Why? It is because carbon containing compounds are known as biomolecules, and there is a good amount of biomolecules present in our body. Oxygen only 46% in the earth crust, but we have a lot of oxygen because we use oxygen to breathe. Then nitrogen also, we have a little bit usage of nitrogen because nucleic acids have nitrogenous bases. Nucleotides are composed of nitrogen base: adenine, guanine, thymine, cytosine. So these are the nitrogenous bases. So that is why we have a good percentage of nitrogen also. Sulfur, calcium, magnesium, silicon, these are sodium, these all are used in our body in a trace amount. So that is why they are also present in the human body in trace amount. So the conclusion from this table, we find out that all the substances which are present in the earth's crust are also present in the human body. This is just the conclusion. Now let us move on to the last topic of the video, that is metabolites. So let us see. So now we are going to discuss about metabolites, primary metabolites and secondary metabolites. Before understanding metabolites, we have to learn about metabolism. So we have the term metabolism. This metabolism is actually known as the sum of all the reactions in human body. Sum of all the reactions. There are two types of reactions mostly in human body on a brief view. One is breaking down and one is forming reactions. When two simple substances combine to form a larger substance, that is known as combination reaction, or like not combination, formation reaction, and that is known as anabolism. And when a larger substance is broken down into simpler substances, that is known as catabolism. So we have anabolism and then we have catabolism. What is anabolism? Anabolism means forming large components from simple components. This is known as anabolism, and forming simple components from larger ones is known as catabolism. Now, anabolism is known as building reactions and catabolisms are known as breakdown reactions. Anabolism plus catabolism, that is, anabolism means plus and catabolism means minus. So the sum of these both is known as the metabolism. Now, after anabolism, we are forming large substances, right? In catabolism, we are forming simple substances, right? So the formation of large substance or simple substances, these are known as metabolites. What are metabolites? So this is about metabolism. Now we are discussing about metabolites. Metabolites are the substances formed after metabolism. What are metabolites? Substances formed after metabolism. Now these metabolites are of two types: primary metabolites and secondary metabolites. So we understood what is metabolism. The sum of breaking down and building up reactions is known as metabolism, or the sum of all the chemical activity inside the human body is known as metabolism. It is of two parts, catabolism and anabolism. Catabolism means breaking down of a larger component to a smaller component. Anabolism means building up a larger components from its constituent smaller components. Suppose we have glucose. We are combining large amount of glucose to form a polysaccharide. That is known as a anabolism. We are taking a polysaccharide and we are breaking down into simple glucose. That is known as catabolism. And metabolites are the substances which are formed after metabolism. Now, what is the meaning of primary metabolism? Let me write it down separately. So, primary metabolites. Primary metabolites are also known as 1 degree metabolites. 1 degree means primary. Primary metabolites are the substances, or actually rather than substances, the metabolites formed which are used or are essential for the body. These are known as primary metabolites. The metabolites which are used or they are essential for the body, they are known as primary metabolites. So for example, we have hemoglobin. For example, we can suppose glucose, etc. So these are the substances which are used, they are essential. So there are some components which when are formed, they are used up. So these are known as primary metabolites. Secondary metabolites on the other hand are 2 degree metabolites. So these metabolites are of unknown used. It means that secondary metabolites are some byproducts or some components which are formed. See, reactions are necessary in our body. Some reactions, because reactions consume energy, reactions are important. Some reactions will have some components or some outputs. So if the output is used up, if the output is important, they are known as primary metabolites. Now there are some metabolites which are discovered, but we don't know what is the use of them. They are not important to the organism itself. First of all, they are not in human beings. So we can say not in animals. Animals do not form secondary metabolites according to this. And then they are not in animals. They are produced by plants, funguses. They are ecologically and economically important. So what are the examples? For example, latex. For example, carotenoids. For example, drugs. These are the substances which are produced by the organism. For example, you take latex, for example, carotenoids. These are produced by the organisms, but they are not of its own use, or else they might be in use, but they are not discovered yet. So they are known as secondary metabolites. They are not in animals. They are produced by plants, fungi, etc. They are ecologically or economically important. Example, latex, carotenoids, drugs, etc. Now let us look at another table, which is the last table, in which we will be discussing about some important secondary metabolites, and then we end the session. Let's see. So over here we have some secondary metabolites written. This is the last part of today's video. So first of all, we have the pigments. So what are pigments? Pigments are the substances which provide color to the plants. So there are some pigments such as carotenoids and anthocyanins which are secondary metabolites. Then we have alkaloids which are morphine and codeine. Terpenoids are monoterpenes and diterpenes. Essential oils which is lemongrass oil. Toxins such as abrin and ricin. Lectins such as concavenin A. Drugs such as vinblastine curcumin, and polymeric substance such as rubber gum cellulose. These are the things whose functions are yet to be discovered by the people. So that is why they are coming under secondary metabolites. So actually over here they have written polymeric substances as cellulose. But I am not sure why they have written over here cellulose, because cellulose is the cell wall. Cell wall of a plants are made up of cellulose. So I don't know why is it under polymeric substances, but still it is given. So it is under the secondary metabolites. Alkaloids are also some kind of drugs. There are some special branches of drugs. Terpenoids are monoterpenes and diterpenes. Essential oils are some oils which are produced by the plants which the plant does not use itself. Example, lemongrass oil. This is not something related to lemon and grass, but it is something else. Toxins are some toxic substances such as aspirin, ricin. Lectins are concavenin A. Drugs which are vinblastine and curcumin. Drugs are also not used by the plant themselves, or else they might be used, we don't know about them. But they are ecologically important, economically important. They give money. And then polymeric substance, rubber gum. Polymeric substances means monomers form polymers. Monomers means small substances form large substances. These polymers such as rubber. This rubber is what we are talking about, latex. Gum cellulose. If you take some kind rubber trees, if you cut them, the rubber comes oozing out. This is called as latex. That latex will form rubber. This latex will form gum, and then you have cellulose. So this was today's video about the differences, or today's video we have discussed about biomolecules. In today's video we discussed what are biomolecules, chemical composition analysis, ASH analysis test. Then we learned about metabolism, catabolism, anabolism, and then we learned about the metabolites, primary metabolites, secondary metabolites, and these are the secondary metabolites. So I hope you have enjoyed today's video. If you have enjoyed this video, do like this video, subscribe to this channel if you are new here. See you next time in the next video. Thank you and goodbye.